The fall webworm Hyphantria cunea is a polyphagous pest that feeds on deciduous tree leaves and causes ecological and economic impact. Behavioral, physiological, and ecological adaptations present in this species are believed to contribute to its rapid spread. In this research, the Hyphantria cunea genome are sequenced to provide information on the phylogeny, evolution, gene function, and gene adaptation. The metagenomic analysis performed for better understanding of its spread and potential gene target for developing new methods to manage this pest. For methodology, collection of sample DNA, colony is collected from single egg mass. Genomic DNA is then extracted using ctAB method. Genome sequencing and assembly. Genome sequencing is performed on PacBio and Illumini, followed by the removal of sequencing adapter and the low-quality sequencing reads. The genome assembly by WTD-BG is evaluated and efficiency of alignment were calculated. Repeat and non-coding RNAs. A repeat library of 4 webworm genome were constructed. Homologous repeat identified by referring specific repetitive sequence database. tRNA scan were used to search for tRNA coding sequence, while rRNA and microRNA were identified based on RFEM database and MIR based database. GeneBlast were applied to identify pseudogene and GeneWise were performed to search for immature termination and frame shift mutation of pseudogene. Gene prediction and functional annotation. The homologous-based gene prediction GOMMA is used to complete homology. The annealed transcriptome was complete by PASA. Reference transcriptome assembly was performed and gene prediction were performed. Finally, all the results were integrated by EVN and annotated by PASA. Gene function were assigned according to BLAST alignment match. Gene family expansion or contraction. CAFE is used to examine the expansion and contraction of gene family. By comparing divergence size and species size calculated could determine whether expansion occur. When the divergence size is smaller than the species size, the gene family is expanding. Positive selection analysis. A branch site model in PAML were used to identify the gene with positively selected site in the 4 webworm genome. Then LLT was performed to detect positive selection on the foreground branch. Transcriptome analysis of different stage and tissues. RNA sequencing were performed on different development stage and tissues. DEG analysis within sample groups were performed, and then the FDR were performed to remove the false positive database. Metagenomic sequencing and analysis. Detail of the gut microflora were obtained by metagenomic sequencing and analysis. Taxonomic identification, relative abundance calculation, and initial gene prediction were performed. All genes in the non-reductant database were translated into amino acid sequence and aligned to relative database. RNA interference with silk fibrin gene. Gene encoding structure protein were silenced by RNAi. RNAi were performed by injecting the corresponding gene-specific double-stranded RNA. Larvae were injected with target double-stranded into the abdomen, and the effect of RNAi were examined by RT-qPCR, and the result were analyzed. The silk web produced collect is rolled into a ball, and diameter of the silk ball were used to calculate the silk quantity produced. For result, high-quality genome of Hyphantria cunea was obtained. The CAFE analysis shows some chemosensory and detoxification genes such as CSP, CCE, GST, and UGT were expanded. Transcriptome analysis showed the most chemosensory genes were expressed in the antenna, while most detoxification genes were expressed during feeding peak. Many nutrient-related genes and one detoxification gene, H P450, were under positive selection, suggesting crucial role of this gene in host adaptation. At metagenomic level, several microbiome communities in the Hyphantria cunea guts and their metabolic pathway might be beneficial for nutrient metabolism and detoxification and might contribute to its host adaptation. Silk web-related genes were identified from the genome, and silencing of silk protein gene H result in significant decrease in silk web production. As for discussion, the CSP, CCE, GST, and UGT gene family were expanded in Hyphantria cunea. GST and CCE mediated insect tolerance to allele chemicals and resistant to insecticides. UGT involved in detoxification and regulation of symbiotic in insects. CSP were grouped together and expressed in the antenna, suggesting role of olfactory, sensory, and host location. Web of 4 webworm provide shelter for larva development. The physiochemical characteristics of the silk web are modified by the silk protein. Silk protein gene was selected to RNAi test to explore the function of silk structure protein. The result of RNAi suggests that H-gene fit H play critical role and could potentially be used as a target for future pest management. As conclusion, this finding explain the host and environmental adaptation of Hyphantria cunea at the genetic level and provide partial evidence for the cause of its rapid spread and potential gene target for innovative pest management strategies.